A large pharmaceutical company such as GlaxoSmithKline will make and test for drug activity many thousands of organic compounds each year. For each compound that is eventually marketed as a medicine, perhaps 10,000 compounds will have been made but found unsuitable in some way. Many drug molecules work by binding to an enzyme or to a receptor molecule on the outside of a cell. Such compounds can be quickly screened for biological activity using robots to measure how well the potential drug binds to the enzyme or receptor. This type of screening uses isolated enzymes and cells and requires only a few micrograms of compound. Pharmaceutical companies now use a number of techniques, collectively called combinatorial chemistry, that allows a few milligrams of many related compounds to be made. Chemists use the word synthesized. These are computer control syringes that add reagents and solvents and carry out other operations in a pre-programmed sequence. In this machine, the syringes are here. It has 4 blocks, each with 96 reaction vessels. The reagents and solvents are stored in these containers. The 96 reaction vessels on this Teflon block are arranged in a 12x8 array. 96 different esters, for example, could be made from 12 carboxylic acids and 8 alcohols. The base of each vessel, made of porous Teflon, is called a frit. The vessel is filled with reaction mixture by the syringe. Emptying is done either by sucking out the liquid under vacuum or by forcing it out under nitrogen pressure. Scientists determine the type of compound that they hope will have the medicinal effect they require, blocking the action of an enzyme or binding to a receptor, for example. Synthetic chemists work out a sequence of reactions that will produce the desired type of compound. The combinatorial chemists program the robot to carry out an appropriate sequence of operations, adding reagents, heating, agitating, washing with solvents, and so on. These particular instructions tell the instrument what solvents to use. A typical run of the machine can take around 12 hours and can be done overnight. It'll produce a library of 384 compounds with similar chemical structures. Combinatorial chemistry is often carried out using beads made from polystyrene resin. This is called solid phase chemistry. The resin is pipetted into the wells as a slurry with a liquid that is then sucked out. The resin has functional groups that allow the starting material to be bonded to it. The new molecule is built up in stages, but remains attached to the resin so that there's no chance of any of it being lost. At the end of the reaction sequence, the products are released, cleaved, from the resin, usually using a strong acid. At the end of the reaction sequence, the compounds are handled in plates like this. Each plate contains an array of 96 different compounds. These can be sent off for chemical analysis and biological testing, still on the standard 96-well plates that are accepted by all the instruments. After many compounds have been prepared by combinatorial chemistry, a promising compound may be identified. This is called a lead compound. Compounds related to this are then made on a larger scale to identify the most effective compound. This process is called lead optimization, and the technique used is called parallel synthesis. The apparatus shown can be used to make compounds in quantities of between 10 milligrams and 100 milligrams. Parallel synthesis can replicate all the techniques that are possible using conventional apparatus with ground glass joints, adding reagents, stirring, heating, refluxing, evaporating solvents, and so on. However, it is quicker because operations are carried out in parallel and related compounds are made in arrays of 24 rather than individually. The most promising compounds that are identified after parallel synthesis will be made on successively larger scales for further trials of effectiveness, side effects, clinical trial and registration as an approved drug. The final stage before actual manufacture is the pilot plant, in which quantities of up to 100 kilograms can be made. The whole process of discovery of a drug and developing it into a medicine typically takes 10 years and can cost up to £450 million.